Green Ann received her undergraduate degree from University of Southern California and completed her PhD at Stanford University in the lab of Professor Caroline Bartosi. And just as a reminder, Caroline Bartosi was really our opening speaker for this webinar series. So it's great great to have you here to continue this story. And in the Bartosi lab, Green developed and elucidated the mechanism of the lysosome targeting chimeras to degrade extracellular and membrane proteins. She will soon start her postdoctoral fellow in Professor David Baker lab in the Institute of Protein Design in University of Washington. Thank you very much for accepting our invitation, and we are very much looking forward to your seminar.
Thank you for the kind introduction, and thank you to all the organizers for inviting me to this seminar series. Today, I'll be talking about some of the unpublished work that I've done as a grad student in the Bartosi lab. So as shown by all the exciting talks from this seminar series, PROTACs and molecular glues have been very promising for degrading intracellular proteins, and they have inspired our lab to think about how we could address proteins that are outside of the cell, or proteins that lack accessible cytosolic domains. So our goal is to target extracellular and membrane proteome, and why we're interested in these proteins is that 40% of protein encoding genes are extracellular and membrane proteins, and there are several bad actors in these classes of proteins as well. So for example, for secreted proteins, we have cytokines, immune complexes, and aggregates that are driving many different diseases. For membrane proteins, we have receptor tyrosine kinases, integrins, and checkpoint proteins as examples. So the proteasome has worked well for degrading intracellular proteins, but lysosome is another cellular machinery where proteins get degraded. So our hypothesis is that we can take advantage of an endogenous machinery that can bring these outside proteins to the inside of the cell and traffic them to the lysosome for degradation. And using this outside-in strategy, our degraders don't need to be cell permeable, making them much more flexible in design. So with that in mind, we developed LYTACs, or lysosome targeting chimeras. And these molecules harness an endogenous lysosome trafficking receptor. In this first iteration of our LYTACs, we harnessed CIM6PR, or cation independent mannose-6-phosphate receptor, which normally binds to lysosomal hydrolases with mannose-6-phosphate glycan and traffics them to the lysosome. And this receptor is known to recycle between the cell surface, Golgi, and endosomes, and release their cargoes into lysosomes. So LYTACs are consisted of two components. One is the target binder, that could be an antibody or small molecule, and another side is the M6PR binder. So we chemically synthesize a ligand consisted of mannose-6-phosphonate glycopeptides that can engage the cell surface mannose-6-phosphate receptor. And so our LYTACs can form a bridge between the receptor on the cell surface and the target protein and drag the protein to the lysosome for degradation. And so using this approach, we showed degradation of several membrane proteins and secreted proteins. So here I'm showing you one example where we degraded EGFR, which is a membrane protein that's overexpressed in many different cancer cells. And we use cetuximab, which is an antibody against EGFR, conjugated to our mannose-6-phosphonate glycopeptides, making it cetuximab M6PN or cetuximab LYTAC. And with our LYTAC molecule, we showed degradation of EGFR in HeLa cells. And so we demonstrated proof of concept through this first generation of LYTAC. And following the development of our LYTACs, there's been a lot of interest in developing extracellular degraders outside of our lab in both academia and industry. So recently, there's been these other molecules called ABTACs, PROTACs, and KINTACs that degrade membrane proteins. And these have been very exciting because we can really start to expand the scope of degradable targets. But one of the challenges across all of these platforms is that membrane protein degradation efficacy is pretty variable across different targets and cell lines. And as of now, we don't really understand why there is this variability, especially for membrane protein degradation. And to expand these technologies as therapeutics, we want these to be generalizable, or we want to have a way to predict where and when it's going to work well. And so we were really inspired by the mechanistic work on PROTACs and molecular glues that really helped advance those technologies. And so we hope to better understand the mechanisms of these extracellular degraders as well. So those are the questions that we wanted to ask what makes these extracellular degraders successful, and what might be preventing degradation. And we want to specifically focus on membrane proteins, because we like to think of these membrane protein degradation separately from degradation of soluble or secreted proteins. And so from our first LYTAC work, we did do a CRISPR screen where we discovered genetic factors that regulate internalization of soluble cargo. And in general, this internalization of secreted proteins or soluble cargo is correlated with the expression level of M6PR receptor. But for degradation of membrane protein, we find that it is not correlated with the receptor expression. And we also do not see a correlation between the uptake of soluble cargo and membrane protein degradation. So for example, we compared 5 different cell lines here. So for K562, we see significant uptake of a fluorophore labeled cargo with our LYTAC molecule. But when we try to degrade EGFR in the same cell line, we barely see any degradation. So we were convinced that the factors that are driving membrane protein degradation are pretty complex and different from the ones that are driving internalization of a soluble cargo. And so to determine the genetic factors that drive the degradation of a membrane protein with our LYTACs, we conducted a genome-wide CRISPR knockout screen where we took these cell lines that express EGFR and transduced them with a genome-wide CRISPR knockout library. And we treated these cells either with cetuximab or cetuximab LYTAC. And with these cells, for our phenotypic sort, we turned to magnetic-based cell sorting for rapid isolation of bound and unbound fraction. So for the magnetic sort, we utilized protein A magnetic beads. Protein A is known to bind to the Fc region of human antibodies. So they should bind to both cetuximab and cetuximab LYTAC. And so for the cells that still express EGFR on the cell surface, cetuximab or cetuximab LYTAC would bind to those receptors, and we can isolate them with protein A magnetic beads. And so when we knock out a gene that's essential for LYTAC activity, we should no longer see LYTAC-mediated degradation, and those guide RNAs would be overrepresented in the bound fraction. And so we collected both the bound and unbound fraction here and performed sequencing. And this was in collaboration with Aurora Camber from the Basic Lab and Simon Wisnowski. And we saw several different hits that both had positive effect or negative effect. So what that means is that for positive effect, I'm showing them in blue, where these are the genes that are normally required for degradation. So if you knock out these genes, we would lose LYTAC activity. Whereas for the genes that had negative effect, shown in red, these are the genes that normally prevent degradation. And if we knock these out, we can potentially improve LYTAC activity. And so we were first very pleased to see that CIM6PR was one of our top positive effect hits. And this was expected because our LYTACs need to bind to this receptor for lysosomal trafficking. But to understand what all of these other hits mean, we first wanted to visualize them and sort of compartmentalize them into different cellular pathways. So we did observe that many of the hits are involved in endolysosomal pathways. So as shown here for clathrin-coated pit, early endosome, late endosome, and lysosome. And this was sort of expected given the endolysosomal trafficking of the mannose-6-phosphate receptor. But for this work, we wanted to focus on the features outside of this expected pathway that define the success of degradation events by our LYTACs. And so we wanted to focus on this NEDDylation pathway, retromer complex, and the M6P biosynthesis pathway in the Golgi and ER. And for today's talk, I'll sort of just give a brief overview of two of these pathways and how they're important for LYTAC-mediated degradation of a membrane protein. So starting with the NEDDylation pathway, I think the audience here would be very familiar with this, but just to briefly go over, NEDDylation is required to activate an E3 ligase. So in our case, our E3 ligase was colin-3 or COL3. And NED8 is a ubiquitin-like molecule that gets transferred from E1 to E2 to E3 complex. And once E3 ligase is NEDDylated, it becomes activated, which means that it can go ahead and ubiquitinate its substrate protein. And interestingly, all of these components that NEDDylate COL3, including COL3, were shown as positive effect hits from our CRISPR screen. So we first wanted to confirm this result and make sure that all of these components are required for LYTAC activity. So we knocked out a couple of different components in the COL3 NEDDylation pathway and saw that LYTAC-mediated degradation of EGFR was ablated when we knocked out these genes, confirming our CRISPR screen result. We also looked at another way to perturb NEDDylation by using a chemical inhibitor of NEDDylation called MLN4924. And we also observed that our LYTAC activity was sort of prevented when we used this NEDDylation inhibitor. And so it seemed that NEDDylation of COL3 is essential for LYTAC activity, but we wanted to understand where in the degradation pathway is COL3 playing a role and how it's affecting LYTAC activity. So we first looked at if COL3 is changing the surface expression of our receptor M6PR. And we observed that there's no difference between the wild type and COL3 knockout cells in terms of their cell surface M6PR expression level. Then we wanted to ask if COL3 is changing the internalization rate once our LYTAC engages the target and the receptor. So here we looked at internalization using a soluble cargo, which is a fluorophore-labeled antibody. And we also did not observe a change in internalization between the wild type and COL3 knockout cells. And so this showed us that COL3 might be playing a role that's important for post-internalization transfer to late endosomes. And so we wanted to sort of confirm that by visualizing EGFR. So these first two columns are showing wild type and COL3 knockout cells that are untreated. So when they're untreated, EGFR is mostly on the cell surface. But when we treat with our LYTAC molecules for extended period of time, 72 hours, in our wild type cells, most of EGFR is gone. Whereas in COL3 knockout cells, EGFR seems to accumulate in these acidic compartments that co-localize with LAMP1, which is a marker for late endosome and lysosome. And so we also looked at where LYTACs are located in COL3 knockout cells after treatment. And we observed that in wild type cells, most of the LYTACs are gone similar to EGFR, but in COL3 knockout cells, LYTACs also accumulate in these vesicles that co-localize with M6PR, which is normally utilized as a late endosomal marker. So it seemed that our EGFR LYTAC complex are accumulated in acidic compartments in COL3 knockout cells. And so we wanted to understand how COL3 is regulating endosomal maturation. And so to get at that question, we first wanted to see what COL3 interacts with. And specifically, we wanted to look at activated versus deactivated COL3, since activated COL3 needs to be NEDDylated. So we performed COL3 IP proteomics in cells that were treated with either DMSO or the NEDDylation inhibitor. And we identified this protein called sequestosome 1 or SQSTM1 that interacts with the activated form of COL3 when we did not treat with the NEDDylation inhibitor. We also performed ubiquitin enrichment proteomics in wild type and COL3 knockout cells. And we also saw this same protein, sequestosome 1, that is ubiquitinated less in the COL3 knockout cells, which implies that COL3 normally ubiquitinates sequestosome 1. And so sequestosome 1 is normally an autophagy receptor, but it's also known to be a scaffolding protein whose ubiquitination is known to be involved in endosomal maturation, usually by other E3 ligases. And we wanted to confirm that sequestosome 1 is also essential for LYTAC activity. So we knocked out sequestosome 1 and validated that our LYTAC activity is reduced. And similar to COL3 knockout, when we look at EGFR localization after extended period of treatment, we see accumulation of EGFR in these acidic compartments. And so NEDDylation of COL3 ubiquitinates sequestosome 1, and ubiquitinated sequestosome 1 seems to be needed for endosomal maturation of lysosomes, or endosomal maturation to lysosomes. And given the critical role of COL3 NEDDylation for LYTAC activity, we wondered if the expression of NEDDylated COL3 can be correlated with degradation efficacy. So we compared the expression levels of NEDDylated COL3 and degradation ability across 11 different cell lines spanning 8 different tissues. And we observed that the cell lines that had higher degree of NEDDylated COL3 showed higher degradation efficacy of EGFR. And so this correlation suggests that NEDDylated COL3 is a predictive marker for LYTAC-mediated membrane protein degradation. And so moving on to the next cluster of genes, which had negative effects, which again means that these are the genes that are normally preventing LYTAC activity. And so if we were to perturb these genes, you could potentially enhance LYTAC activity. And so all of these genes were part of this N-glycan M6P biosynthesis pathway. And so lysosomal hydrolases are synthesized in the ER, where they get tagged with the mannose-6-phosphate glycan as they're transferred to the Golgi. And once these enzymes are tagged with mannose-6-phosphate, they can bind to the mannose-6-phosphate receptor. And this complex can either traffic back to the cell surface or traffic to the endosome, where they release their cargo to lysosomes. And all of these glycosyl transferases shown here in red that are responsible for putting on this mannose-6-phosphate glycan on lysosomal hydrolases were shown as negative effect hits from our CRISPR screen. And so we first wanted to confirm that knocking these genes out would improve LYTAC activity. So we first knocked out ALG12 and GMP-TAB, and at an earlier time point, we did observe greater or enhanced rate of lysosomal trafficking of our targets, EGFR. And we also saw this across different membrane protein targets, such as c-MET and CA9 as well. So we wanted to understand how these M6P biosynthesis genes are regulating LYTAC activity. So again, we wanted to see if it changes the internalization of our LYTAC complex. And when we looked at wild type cells and these knockout cells, we observed increased internalization in these knockout cells. And so initially from this result, we assumed that these ALG12 or GMP-TAP might be regulating the surface expression of mannose-6-phosphate receptor. So it was possible that knocking out these genes could increase the number of receptors on the cell surface. But when we evaluated the cell surface expression level of CIM6PR, we did not see a significant difference across the wild type and knockout cells. And so at this point, we sort of had a hypothesis of what might be happening. So to sort of go over again, these lysosomal hydrolases are synthesized in the ER, and they get transferred to the Golgi, where they get tagged with the M6P glycan, which then can bind to the receptors that can traffic back to cell surface or the endosome. But when we knock out these M6P biosynthesis genes, we're basically preventing this process from occurring, which means that a lot of these mannose-6-phosphate receptors are unoccupied with endogenous lysosomal proteins. So that could open up more available mannose-6-phosphate receptors on the cell surface that LYTACs can engage. And so our hypothesis was that knocking out these biosynthesis genes could lead to more unoccupied CIM6PR on the cell surface, and that could lead to better lysosomal trafficking. And so to validate this hypothesis, we first wanted to see if there's higher binding of our LYTACs in these knockout cells. And so we conducted a binding assay where we incubated cells on ice with our LYTAC molecules. And we did observe that in these knockout cells, we saw increased binding of our LYTACs compared to the wild type cells. And these bindings were ablated when we utilized M6P inhibitor, which shows that the binding is M6PR dependent. And so this result implied that a significant fraction of the receptors are occupied by endogenous lysosomal proteins, and that and we wanted to really validate that that's the case and identify what are some of the lysosomal proteins that are occupying these receptors. So to do so, we performed quantitative cell surface proteomics, where we biotinylated the cell surface on ice and enriched for the biotinylated proteins and performed quantitative cell surface proteomics and compared the wild type and the GMP knockout cells. And what we observed was that we saw downregulation of certain proteins on the surface of knockout cells. And these proteins were lysosomal proteins that are previously characterized to be modified with mannose-6-phosphate glycans. And we also confirmed this by cell surface staining by flow cytometry, and saw that there's reduced cell surface lysosomal proteins in these knockout cells. And so we confirmed that knocking out the M6P biosynthesis genes results in more unoccupied receptors on the cell surface. And we discovered that a significant fraction of cell surface CIM6PR remains occupied by endogenous M6P modified glycoproteins. And through our unbiased mechanistic study here, to uncover the drivers of extracellular degradation, we revealed fundamental insights regarding receptor occupancy and trafficking. And the study can elucidate new design principles for lysosome-targeted protein degraders. And the findings from this studies have direct implications for other therapeutic modalities. So for example, enzyme replacement therapy for lysosomal storage disorder relies on CIM6PR trafficking. So they usually generate recombinant M6P tagged lysosomal enzymes to deliver to lysosomes. And our findings in this study on the occupancy of CIM6PR will be important for this type of therapy. It can also have implications for other therapeutics that could utilize M6PR for internalization, such as ADCs and oligonucleotides as well. And finally, the genomic screening approach that we utilize here using the protein A magnetic enrichment can be applied to other extracellular antibody-based degraders to study mechanistic principles of these membrane protein degraders and other lysosome trafficking receptors. Okay, so that leads to, I would like to thank my advisors, Carolyn and Steven. So Carolyn has really given me a lot of freedom over the years and has been an amazing advisor for the last 6 years. Steven has led the pioneering work on LYTAC, and he's been an incredible mentor to me over the last 6 years as well, and advised me on this project. I'd like to thank our collaborators, Dr. Nick Riley, who has done all the proteomics work that I presented, Dr. Simon Wisnowski and Roy Camber, and Professor Basic for their advice on the CRISPR screen, and Salvador, my rotation student, for his support. And I'd like to thank the rest of the Bertuzzi lab and my funding sources, and I'd be happy to take any questions.